Hi, everyone. Thanks so much for joining us today. I'm Irene Song, Senior Director of Global Product at Packaging Biotech, and I'm really happy to welcome you to this exciting webinar. Before we get started, I would like to quickly introduce who we are at Packaging. So we are a global CRO and CDMO focused on custom AAV production, along with lentivirus, mRNA, and plasmid DNA manufacturing. Our goal is to help speed up gene and cell therapy development by delivering high-quality, scalable vector solutions. We support our partners all the way from early discovery to clinical and even commercial stages. I'm really thrilled to introduce today's speaker, Dr. Franck Kalume. Dr. Kalume is a principal investigator at the Center for Integrative Brain Research at Dr. Kalume is a principal investigator at the Center for Integrative Brain Research at Seattle Children's Research Institute, and he's also an associate professor at the University of Washington in the departments of neurological surgery, pharmacology, and global health. His research is on understanding the cellular and the molecular mechanism behind epilepsy, especially Dravet syndrome. He's made some amazing contributions to how we understand this disease and how we might treat it. Today, he's going to walk us through some exciting work on the dual-AAV gene therapy approach that is showing promising in correcting Dravet syndrome in mouse model, which is a big step forward in tackling one of the toughest pediatric epilepsies. If you have any question during the talk, please feel free to type them into the little speech bubble icon that you will see on the upper left corner of your auditorium page. And we will collect your question and get them during the Q&A session at the end. And with that, I'll hand it over to Dr. Kalume.
All right. Thank you, Irene, and the team at Packaging for this invitation to present in this webinar with all of you. And I'm sorry for the early technical challenges. It is my excitement this morning. So I'll start over and just again say my apology for the technical difficulty here, and I hope that you can hear me without any problem at this point. But so I'm excited this morning to present to you our work on gene therapy in Dravet syndrome. And I should mention that this work has been is a continuation of a lot of the efforts that have been conducted in my lab for decades. And okay, there we are good. And for decades, as I was saying, and this started out with our understanding of the Dravet syndrome using a mouse model. And today we are hoping to have some insight in how to treat them, and moving all the way to clinical therapy or to the bench side is our main goal. And so with that, I would like, before I talk about all of this, I would like to start with disclosures. And here I should mention that we have 3 patents on the technologies that are being used in this particular work that I'm going to present to you. And also we have funded a startup company with colleagues from the Allen Institute who are my collaborator in this journey. And so as an outline of this presentation, I would like to tell you that I'm going to start with an introduction that will speak briefly about Dravet syndrome, go into its etiology and pathophysiology, and then talk about the hypothesis that led to all of this work. And then from there, we'll look at the way we characterize the vectors that are our therapeutic vectors in vitro, and how we move them into into in vivo setting in our mice and evaluate two type of vectors. One is the one targeting the inhibitory neuron specifically, and the other is more pan-neuronal in its characteristic. And then from there, we'll go to a conclusion, and I hope I will have enough time to answer your questions at the end. So with that, so what is Dravet syndrome? And many of you in the audience may know about Dravet syndrome. This is a rare but very devastating form of epilepsy. It's belonged to a class of severe form of epilepsy known as developmental epileptic encephalopathy. And this is a designation that is associated, that is referred to for a group of epilepsy known to be very severe with the seizures and interictal epileptiform event contributing to the cognitive and behavioral impairment that are observed in these patients. The incidence of Dravet syndrome is estimated to 1 in 20 to 1 in 40,000 births. And together, all the Dravet cases represent about 7% of all severe form of epilepsy that start before the age of 3. And at the bottom of this slide, you can see that I'm presenting to you some faces of kids with Dravet syndrome, some of whom that are met over the years at family conferences, and they are a great reminder of why we are doing this work and how we are motivated to try to make life a little bit better for them. And so we know from genetic studies that Dravet is primarily caused by monoallelic or heterozygous loss of function mutation in SCN1A. And this is the gene that encodes the alpha subunit of the sodium channel, as shown here. And this is the part that forms the pore of the channel. This association with this mutation started out with a very interesting work that was conducted by the group led by Peter de Jong in Belgium almost 25 years ago. And we know that about 80% of cases with Dravet are associated with this mutation. However, we also know that there are several other genes illustrated in this slide that are associated with Dravet-like phenotype, and those represent a small portion of the cases. And the clinical manifestation of Dravet is very well characterized. It was first characterized in 1978 by Dr. Dravet, shown in the picture with me here on the slides. And unfortunately, Dr. Dravet is not with us anymore. She passed away early this year, and I would like to pay a tribute to her. She was a very humble and giant in this field, and she helped really move the research forward, and also patients deal with Dravet syndrome. She managed many kids with this syndrome. So briefly, the classical phenotype of Dravet syndrome is marked by onset within the first year of age in a child who appear otherwise normal. And the first seizures that appear are usually triggered by elevation of body temperature or in the context of fever. But not only fever can trigger these seizures, this can also occur in the context of vaccination and or even just a mere elevation of temperature during a warm day. And so after these first seizures, kids with Dravet will experience multiple other seizures of different type, including status epilepticus. And as the disease progresses, several other comorbid conditions will appear, and these include cognitive, motor, behavioral, and sleep and circadian rhythm disturbances. And very sadly, is we know that kids with Dravet are at a higher risk of premature death, and this can occur due to sudden unexpected death, status epilepticus complication from status epilepticus, or even from accident. And this is a big concern for family and all the people who are involved in taking care of kids with Dravet. Very relevant for our talk. We know that despite the availability of many different types of anti-seizure medicine on the market, and even some that have been developed recently, such as fenfluramine and Epidiolex, kids with Dravet are very resistant to most of the anti-epileptic drugs. And in most of the cases, there is only partial alleviation of the symptoms. The seizures continue to appear, multiple comorbidities that remain present, and the side effect due to polytherapy, which is often the strategy used in management of this disease, are still present, and we have reduced quality of life in most of the kids, and they are all still at risk of sudden unexpected death in epilepsy. So as you can see, there remain a very significant unmet medical need to develop and characterize effective treatment for this syndrome. There are several groups around the world that are working just on this, trying to develop new therapeutic strategies for Dravet syndrome. And among them, those that are getting most of the attentions are those using innovative genetic approaches such as CRISPR and more that are being developed in many other labs, including ours. Our work started out, as I said, almost 2 decades ago, when I started as a postdoc in the laboratory of Dr. Catterall, here pictured to the top of this slide with me. And I would like to also give homage to Dr. Catterall, who was a pioneer in the field of ion channel, and who passed away almost a year ago, or over a year ago. And coming back to this research, as we started out, we developed a mouse model of Dravet syndrome by deleting a portion of the gene that that express this part of the channel from transmembrane segment 3 in the domain 4 of the channel, all the way to the C terminus of this protein. And we characterize this mouse model and show that it expresses a lot of the phenotype observed in human patients. And we conducted a series of studies that I'm not going to go through all of them here, but I'll mention that through these studies, we discover several insights into the pathophysiology of Dravet. And here I'll focus only on 2 that are very relevant to this study. One is that we discovered that Dravet causing SCN1A mutation leads to reduced sodium current and excitability in inhibitory neurons while leaving the excitatory neurons without any disturbance. And 2, we found that the key Dravet syndrome phenotype are recapitulated when we introduced the mutation specifically to GABAergic interneurons of the forebrain. And together, this finding led us to develop what is known now in the field as the interneuron hypothesis for Dravet syndrome pathophysiology. In short, what it says is that Dravet syndrome is primarily a disease of forebrain inhibitory neurons. And I would like to mention that these hypotheses have been expanded upon with new findings from other labs in the field, where they noted that among the different subtype of interneurons, it is the parvalbumin interneurons that play the most critical role. And the second thing that has been observed is that there is a transient developmental window when the inhibitory neurons play a key role in the pathophysiology of the disease. But after that developmental window, there are secondary other manifestations that become to contribute to the phenotype development. I am not going to focus on those, and just go back to our interneuron hypothesis, where we think that because interneurons play a critical role in the mechanism of Dravet syndrome pathophysiology, then uh, they constitute a very valuable target for gene therapy. And we think that a gene therapy that is targeted to these cells will be very efficacious in preventing all the phenotype of Dravet syndrome, and not only in doing so, but also in leading to much less side effect because of the precision that this brings in. So for our work, we had to overcome two main challenges in the field. We wanted to develop an interneuron-specific gene therapy for Dravet. However, there were two main challenges. One is that there was the therapeutic gene, the SCN1A, exceed the capacity of, or the therapeutic gene size exceeds the maximum capacity of AAV, which is estimated about 4.7 kb, whereas the SCN1A coding sequence is about 6 kb. And then the second challenge was that we wanted to target inhibitory neurons specifically, but at the time there was no good promoter that will help us target these easily. Why? It's because the promoter that was available, such as GAT2, was too large to be packaged in AAV by themselves, and let alone to be packaged along with the therapeutic gene. So what I tell my student is that we had a situation where we had a very large machinery that we needed to deliver, a small vehicle, such as the pictured small truck, and we had a very defective GPS system in order to target the destination. So how to overcome these challenges? This is where we team up with our colleague at the Allen Institute. We were working at the time on a project where they were doing RNA-seq studies and discover some enhancer that they could use to label different cell type in the brain and map their location and many other phenotype of those cells. So of interest among some of the enhancers was the DLX enhancer that was shown that it could target inhibitory neurons with high precision, and this is where they show in mice that had inhibitory neurons labeled with the TD tomato fluorescent, and they could send in AAV and label the same these inhibitory neurons. And this finding was consistent with prior finding by the Fischel lab. The Fischel lab is a developmental biology lab where they've been studying the development of inhibitory neurons. And this lab and many others have shown that the DLX enhancer can be used to mark inhibitory neurons. And in this study that they published in 2016, they show that they can use the DLX enhancer to target inhibitory neurons with AAV. And as shown here, inhibitory neurons are marked in red, and the AAV deliver GFP and there is a nice colocalization. So we think that the DLX56 is an interesting system or technology that we could use for our gene therapy. Then there was the second problem. How do we overcome the second problem of trying to deliver a very large gene? And for this, we rely on the technology called the split-intein protein splicing technology. This is a system that is used by small organisms like bacteria and plant in nature. They naturally express uh, when they try to build a large protein, they will use two different genes as shown here. And these genes will express polypeptides, the N and C polypeptide of the protein of interest. And this segment will have an intein, N and C intein attached to them. And this intein will come together, bind together, and join the two polypeptide and excise themselves out of the picture. And this process happened without any enzyme, which is very exciting, and it is very fast type of process. And in our case, we thought this can be used as what we call in the lab affectionately the IKEA-like method, where we could take our gene of interest, SCN1A, split it in half, and package it into two AAVs. And then each of the segment of the gene will express part of the protein with the intein, and then this intein will mediate the junction of the two protein to form the full-length SCN1A and excise themselves out of the way. And can we do this? And we started out our work where we built our construct with a CMV promoter on each. And so these are our two constructs for half of the gene. And then we cultured hex cells and expressed this in this culture system. And what we found out is that only in cases where we had both the N and C terminus segment expressed is when we could get the full sodium current at a maximum shown here, almost at the same size as when we express the SCN1A as a whole, as a full-length channel. But in cases where we only had one of the segment expressed, there was no current. So as you can see here in the statistics is that the full length is showing large current. When we express the two component, we see also a comparably larger current and no current in the other conditions with only a single segment, or when we have the GFP alone expressing, or the beta subunit, which are auxiliary subunit of the sodium channel here. And so that was interesting and very encouraging that our channel can express. And now the next question we wanted to know, can we actually deliver the channel where we want it to go in the inhibitory neurons? And to do so, we built some other constructs. And this now our construct with the DLX 2.0 promote an answer so that we can deliver this into inhibitory neurons. And these were packaged into AAV by our partners at PacGene. And then we deliver these AAV through ICV in the young mice between the age of P0 to P3. And here is immunohistochemistry picture of what's happened. And because each of the segment had HA tag or flag tag, then we could easily identify the protein when it is expressed. And as you can see, there is expression in the forebrain here, showing in both of these slices when we detect them with a flag antibody or HA antibody. And then when we look at a cellular level, we saw that in the case where we had the two segment express, we could pick the HA antibody, the flag antibody, and all of them co-express with GAD, meaning that they were being delivered specifically in inhibitory neurons. And in cases where we had only one of these, we could see that we could pick with the associated tag that is that come with that component. Here for C terminus is the flag, and that also co-label with GABA antibody, and similar things with the N terminus. And so we had very specific expression in inhibitory neurons. But how distributed is this expression across different regions of interest that we would like it to be in? And these regions of interest are those regions that are known to be involved in seizure generation. And so when we look at this, we did this experiment or this analysis where we look at when we have HA or flag expressing, are these all into inhibitory neurons in different brain regions, the visual cortex, the motor cortex, and the hippocampus. And we saw that 100% of these expression happen in inhibitory neurons. But that is good. We see very good specificity. And is this all reaching all of the interneurons in these regions? We saw that that's not all very completely that all the inhibitory neurons are not completely targeted, but we were getting a completeness of targeting that was between 25 to 50% of cells of inhibitory neurons in those regions. And so it's a partial targeting of cells in this region. And we wanted to know, is this sufficient to actually rescue the phenotype of Dravet syndrome? And so to do so, we injected these vectors in young mice again, and then followed them. These are Dravet mice at P25 to P35 at the peak of the phenotype. We tested them for seizure susceptibility using our thermal seizure induction protocol that I'll explain in a little bit. And then at P70, we look at mortality all the way to P70, as you can see in this picture. And so when we look at mortality, we saw that with a lot of excitement that none of the treated mice died. Our control mice, we received either a single vector or control vector, were dying at a comparable level as to the one that were untreated. So this is a very encouraging result. Then we wanted to know, are we actually protecting this from the seizures? And to look at that, we now did the thermal seizure protocol here, where we raise the body temperature of the mice incrementally by 0.5 degree every 2 minutes. And then we stop this protocol whenever the mouse had a seizure or if they reach the temperature of 42 degrees Celsius. And this is our recording system. And we look at two different outcomes of this. We look at myoclonic seizures and generalized tonic-clonic seizures, which are the type of seizures that we have seen in we have shown in our previous studies that occur during this type of testing. And this is an example showing you that with only 2 degrees of elevation in body core temperature, will have a generalized tonic-clonic seizure in the Dravet mice, which is shown here by EEG recording. And very interestingly, this is the type of phenotype that is also seen in the patients. In the study here from 2001 by a Japanese group, they showed that babies with Dravet syndrome, when they are put into a warm bath or warm tub of water, during bathing. And if the temperature rises by a few degrees Celsius, they can trigger a seizure. And so using this protocol, we tested and see how our mice responded upon when they received the treatment. For the myoclonic seizure, we saw that they were protected all the way to 40 degrees Celsius, but it's only for temperature higher than this that they started showing, some of them started showing seizures. So there was a good protection, but it was not complete. And what about for generalized tonic-clonic seizure? We saw an impressive protection all the way to 41.1 degrees Celsius, and only about 10% of the mice will had a seizure at the last temperature point of the experiment. So we thought that this was a very good indication of protection against generalized tonic-clonic seizures that and myoclonic seizure, partial protection, and all of these were done in the the cases where we were triggering the seizures or provoking the seizures. What about spontaneous seizures or spontaneous epileptiform event, which are the type of event that you see in a clinical setting and in vivo? And here, what we saw is that our mice normally they will have interictal spikes, and but when they are treated, we see that there is a much less interictal spike frequency than in the untreated. And similarly, these mice will exhibit myoclonic seizures that can be shown on EEG by this burst of activity in the EEG that are associated with another burst of activity in the EMG recording. And here we see that none of the treated mice show this type of activity on EEG. And a small portion, because these studies that I'm presenting here were only through 8 hours of recording, we saw that a small portion of our mice exhibited generalized tonic-clonic seizure during the time of the experiment, but those that were treated, none of them show any of this type of event. And so together, this is showing us that our treatment is protective against spontaneous epileptiform events that are present in our mouse model. Then to further characterize our treatment or our vectors, we generated a conditional knockout Dravet mouse. And this knockout was generated with only inhibitory neurons of the forebrain carrying the mutation. And to do this, we use the Krelok strategy as shown here in the breeding strategy. And what we were interested in is that we can reproduce a scenario where the disease-causing mutation is present only in the cells that we intend to treat. And so then we can do a precision therapy on these mice. And here I'm showing you that these mice with only in forebrain inhibitory neuron knockout exhibit even a more severe mortality than those with global knockout. And you can see here that we lose all the mice by week 5 postnatally. Then when we inject them with our AAVs and monitor this mortality, we saw that remarkably none of them died all the way as we monitored to week 10. So showing again a very strong protection. And then when we look at the seizures or the terminally induced seizures, we saw that none of the treated mice exhibited these myoclonic seizures or generalized tonic-clonic seizures. So this was very exciting, and it shows to us that with a precise delivery of our gene therapy in the cells that are sick, we can actually rescue all of the phenotype, even in cases where the phenotype is very severe. Then the next thing that was very important for us clinically is that will this last for a long time? And the promise of gene therapy is that you can do one treatment and the individual is protected for the rest of their life. And here we look at some of the mice that we tested and follow them all the way to 1 year of age. And we saw that only a small portion died throughout all of this monitoring period. And then when we tested this from thermally induced seizures, we see that they still have good protection against thermally induced seizures and even better protection against generalized tonic-clonic seizures induced by elevation of temperature. So we think that this has a promise of being uh, one shot and you are done for the rest of your life type of treatment, at least in the mice, and it need to be proven in humans. And then the next question we wanted to ask was that is that important for us to go through all of the very complex approach of targeting inhibitory neurons specifically? Is it if this is necessary, then we will see by comparing the interneuron specific approach with a pan-neuronal approach. And to do this, we made a new AAV vector that are that are driven with the human synapsin 1 promoter. And these were injected in the mice, and we could see a good expression of our vectors. And when we look at inhibitory neurons, we see that we specifically were targeting about half to a third of the inhibitory neurons. As far as excitatory neurons, we were able to target all the excitatory neurons. And the completeness, the percentage of these that we are targeting is only in the range of 30% to somewhere 15%, depending on the region, and for excitatory neurons, almost the same level. Then we followed these mice that were treated, and what we observed was very interesting. Is that unfortunately we saw that these mice exhibited mortality in the prey-weaning period, and we saw that we were losing some of the mice when using a lower dose, which was E to 10 genome copy. We saw a small percentage of the mice were dying, and as we increase the dose to 3 E to 10, we see that the percentage of mice that were dying increased as well. And this indicated that there is a little bit of toxicity of our therapy when we go with the global or pan-neuronal approach. And this is our test for thermally induced seizures. We see that there is also a dose-dependent effect where only the higher dose show good protection on myoclonic seizure, and then a good protection on generalized tonic-clonic seizure. This was encouraging. However, we cannot make a therapy that says, as I tell my students, that if it doesn't kill you, then it will protect you from seizures. Then this is not. We thought that there is a very high downside to going with a global approach. So, in conclusion, what I just showed you is that we generated DLX 2.0 split-intein vectors that are very exciting to us. And we showed that these reconstitute a full-length SCN and NAV1.1 protein in vitro, and that these vectors transduce specifically inhibitory neurons of the telencephalon, or the in the forebrain. And then we also showed that we are getting a strong efficacy against SUDEP, or mortality here, sudden mortality, looking at the terminally induced seizures and even spontaneous seizure event. And then in this case, we did not have any side effect when we're treating the global knockout model. And in the conditional model, we could see that we have complete rescue of the phenotype tested. But when we went with the gene that were pan-neuronal, we saw a good protection against seizures with the higher dose, but that come with very high or severe side effect, high mortality. And so I would like to also mention that we were encouraged by the fact that when we tested these vectors at the Allen Institute as well, in the second mouse model, we were able to see similar finding. And without forgetting that we're getting a durable rescue of the phenotype. So in the future studies, we hope that we can go into evaluating our vector on non-seizure phenotype of Dravet syndrome, which play an important role in the life of patients with this disease. And we also want to look at the durability, which I've shown and finish up some of those studies. And we'll examine the effect of the vectors when administered at a later time point, as this may be the case, will be most likely the case in the clinical scenario where patients will show after symptom development. And we will also evaluate whether there is a point of no return where after the symptom or the disease has progressed to a certain time point, there is no way to rescue those phenotype. And I will end here with giving my thanks to all our collaborators, starting with our many collaborators at the Allen Institute, and principally Dr. Boaz Levy, who is my collaborator there, and with his team and colleagues at the Allen Institute. And this research was funded by an NIH UG3 grant with UH3 with the Brain Initiative. And we have other collaborators such as Viviana Ganuraru on the grant, and Greg Holtritz as well. I'll thank my lab, especially Judy Ru, who did a lot of the work on the characterization of these vectors in my lab. Samantha Henson is a very talented technician in the lab who's continuing with the work in the lab. And we have our collaborator in the Ramirez lab who work with us in the characterization of the expression of the vector. And to the right here, I would like to thank all our funds, so the funding agencies that help us conduct a lot of our work, and of course our partners in the industry in packaging and building up these vectors. And so I would like to end here, and this is just a slide to show you that we have a postdoctoral program for those who are postdoc in the audience that is directed toward helping young postdoc in developing their skill in therapy development. And I'll end here and take questions if there are. Thank you so much.
Okay, thank you so much, Dr. Kalume, for the fascinating presentation. And really appreciate your sharing your work with us. And it's really exciting to see the potential of this dual-AAV approach in treating the Dravet syndrome. Now we would like to open up for questions. So if you haven't already sent your question, you can submit your question by clicking the speech bubble icon in the upper left corner of your auditorium screen, and we will go through as many as we can during this session. So the first question is, in your split-intein dual-AAV system, what are the key challenges in achieving efficient reconstruction, reconstitution, and expression in vivo, and how might this be optimized further?
Yes, that's a great question. And so here, I think we were fortunate that the DLX enhancer was very efficient in sending all the two components, even though they were packaged in two different AAV, into inhibitory neurons. And to our surprise, we saw that whenever one cell received one component, they always receive the second one. So it's not always very obvious that you could receive two components in one cell to make the whole thing. So that's one thing. The second challenge that the second thing that I'll mention is that there is still a challenge because we are only getting a partial rescue, about 30 to 50% of partial targeting, I should say, and 30 to 50% of inhibitory neurons are being targeted. But we do not know if this will work in once we go in human, that we need only a small percentage. But we think that nature or biology is working in our favor here, telling us that we do not need to be perfect. We just need to get this to a certain level where it seems like there is a threshold in order to rescue most of the phenotype. So we'll see how this will translate once we get to human. The challenge here has been mostly to, as I spoke about in the presentation, how to package the larger gene, and this has been very, very difficult to do. And we'll see whether this will remain translatable. All the good stuff will remain translatable once we get to human.
All right, thank you very much.
Nice.
So
Any other question?
Yes. So the next question is, given the promising rescue effects in Dravet syndrome models, what are the major hurdles in scaling this gene therapy approach towards human trials, and how are you planning to address them?
Yes, I think that's a very good question for the next step, is that how do we get this to clinical trial? One of the main hurdle is the size. How do you scale this to a size of a human brain? As you can see, and you know, is that the mouse brain is small, basically the tip of my finger. And when we start talking about a human brain, even for baby, is at a very much larger scale. One of the things that we envision is just how to produce the amount of vector that we need to be able to reach most of the needed areas to be targeted for this therapy. And I think that is something that is big in the field of gene therapy, how to produce this amount of vector needed. We hope that by being very specific to inhibitory neurons, what we'll need is a small amount of vector compared to whether we were compared to the case, for example, if we needed to target the whole brain and all the cells. But that's yet to be demonstrated. We have to do some testing. So we have some of the tests being conducted with our partners at the Allen Institute in non-human primates to see if for a larger brain we'll be able to generate, number one, enough vector, and we'll be able to target all the regions that we need.
Yeah, sounds great. So the next question is, how translatable do you believe these preclinical outcomes are to human patients, especially in the context of variability in mutation types and the timing of diagnosis?
Oh, great. Yeah. So, um, this is a great question. And in developing this therapy, we hope to have a therapy that can be used to most of the patients. And most of the patients, as I mentioned in my presentation, have a loss of function mutation. And this therapeutic approach, regardless of what mutation you have, if the what we know is that the mutation that causes Dravet syndrome leads to haploinsufficiency or reduced number of the sodium channel and the NAV 1.1. So in this case, we think that we can actually, regardless of the mutation, send a corrective or supplement the system with a new, an intact copy of SCN1A and express new protein. Um, so that's the part of the variability um, the diagnosis of the molecular uh, problems in the Dravet. The second part was, can you remind me again?
Yes. So the second part is about, especially in the context of variability, the variability in the mutation type and the timing of diagnosis.
Oh, yes, the timing of diagnosis. And the timing of diagnosis is what I mentioned um, in our future studies, is that we would like to see whether we can rescue uh, uh, the phenotype even after its onset. And the idea here is that if there are secondary phenomenon or secondary changes that happen, it may be too late for us to rescue a Dravet phenotype after it's happened, even with gene therapy. But that needs to be tested. And how far is that after the onset of the disease you can go without and before there is a point of no return? That is something that we would like to test. One of the other things that we discuss a lot with our clinician partners is that for Dravet kids, for example, there may be some of them who would develop uh, who will not be able to speak and they are at the age of 20, for example, especially these days that we have even adult with Dravet syndrome because the management is much better than it used to be. And can we actually rescue something like their ability to speak? And in that case, it may be very challenging. But we hope that we can help their brain into a way that will be possible for them to communicate in some ways and better than they are without the treatment. So that is what we can think of because that's learning a language is a developmental process and it may be at that point a little bit difficult to try to rescue that if the person is already at an adult age.
Yeah, that's true. Especially for the timing, it's probably very critical for restore all these functions. So
Yes, and that's actually one more point I would like to make is that this will be also very important. We hope that it will be important in the field to start identifying those with Dravet as early as possible. Early diagnosis will be very important, and then try to have early treatment on top of that.
yes, that's true. Okay, so we have two questions that are asking about serotypes. So one audience asked, like, could you clarify which serotypes you are using for the gene therapy? Is the vector administered intravenously? And also, what's the vector's biodistribution beyond the neurons?
Yeah, so I'll start with the last question because the answer is that so far we have done biodistribution only in the brain, and our follow-up studies are going. In fact, this morning we were just talking with our core facility about how to do evaluation in outside of the brain and basically throughout the whole body. And the other thing is that what vectors did we use? We used a PHPEB vector, and which is a little bit of an overkill for ICV injection, intracerebral ventricular injection. We know that PHPEB can penetrate the blood-brain barrier, and that's why we used it. And we're thinking at first on treating from peripheral delivery, but we wanted to go into ICV here because we were getting better reproducibility, and with this approach, we are planning to test delivering the same vector from the periphery and see whether we can rescue the phenotype. And as a footnote, we've seen some of our colleagues who delivered the PHPEB from the tail vein in mice and seeing a good biodistribution in the brain.
Thank you so much. I think the other question related to serotype is also probably you already answered part of it, but the audience is also curious, how did you select this serotype? Yeah, I'm also curious, like whether you test a few other serotypes that can also target to the brain.
No, I think we initially I think we tested some AAV9, but then with the idea that we were gonna try to do peripheral injections and all of that, we went with PHPEB.
Yeah. Okay, got it. So the other question, the audience says really lovely work, and do you know why there's not a protective effect against seizure at a higher temperature?
This is more of just, I would say, a speculation. We don't have strong data on this. We think that temperature is a very strong trigger for seizure in the Dravet model, and there is a certain level of protection against thermally induced seizures. And then temperature can overcome whatever threshold we set up to. And the reason that temperature can overcome this, it's maybe just because the targeting efficiency, as we saw, not all inhibitory neurons were protected.
I see. So I think we still have a few other questions, but we are running out of time. We will, send our reply through your email, and hopefully, if you have any questions, feel free to reach to us, and we will help you to forward the questions to Dr. Kalume. And I really appreciate everyone's to join today's webinar. And thanks again for our speaker, Dr. Franck Kalume, for this wonderful talk. And hope like we can see you for other webinars in the coming month. Thank you, and have a great day today.
All right. Thank you very much, Arlene, for coordinating this event. It was a very great pleasure, and thank you to the whole team at PacGen for all their effort. Thank you, and goodbye to the audience, and hope to hear from you if you have any additional question.
Thank you. Bye-bye.
Bye-bye. To present this work.
Sorry, Dr.